Hello everyone, greetings of the day and welcome to week 1, where in the first unit we are going to discuss herbs as raw material. Herbs or plants which are the basis for any therapeutics in a traditional medicine. Let us look what the term herbs mean, what all is included when we say herb. So, let us look at the definition. I am going to introduce to you the different sources from where these herbs can be collected. When these herbs are collected, what kind of different preparations are made from this herb, or what is available in the market, in what form these herbs are available in the market, and other than the therapeutic use, where all these herbs can be used. Majorly they are used for therapeutic from traditional wisdom. So, when we have to define a herb, herb is a plant or part of the plant valued for its medicinal, aromatic, flavoring properties. Typically, it is used as a raw, dried or processed form, simply because the raw, dried and processed form is easier to store and it is more stable. In many preparations or in many cases, these herbs can be used as fresh material also, but we have to take care that if the herbs are collected fresh, then the stability has to be kept in mind. So, in pharmacognosy, where pharmacognosy deals with study of everything that comes from nature, herbs refer to plants or their parts such as leaves, flowers, stems, roots, seed or bark, and sometimes the entire aerial part is used as a herb for therapeutic purposes. They are employed in different preparations of herbal medicines, cosmetics, nutraceuticals and dietary supplements. Classification of herbs when we use them becomes very important, because this helps us to identify a herb, it helps us to know from where we can collect this herb, it helps us to know from where this herb grows majorly, in which part. So, classification is necessary because it helps us to organize these herbs by their uses, properties, life cycles, which makes it easy for us to study, apply and preserve their knowledge effectively. So, classification of herbs is just not an academic, but it is practically very much needed. It ensures that herbs are correctly cultivated, efficiently and studied scientifically, bridging the traditional wisdom with modern applications. So, all this ensures good quality control of the final therapeutic medicine. Is classification very important and does it matter? Yes, of course it matters. Classification of herbs helps us in systematic understanding. Herbs have diverse properties. Some of them heal, some of them flavor food, others decorate space. So, in horticulture, if we see, sometimes the horticulture plants are used as therapeutic medicine, otherwise they are just used to decorate landscapes, gardens and corridors. Classification helps us to provide a structured way to distinguish between medicinal, culinary, aromatic and ornamental herbs. There is ease of application. By grouping herbs according to their usage, practitioners, chefs, gardeners can quickly identify which herb suits a particular need. These days, food is also cooked with scientific knowledge. So, whenever chefs want to design a food for scientific use, suppose there is food for high dissolving lipid or for high cholesterol, food specially designed for diabetic patients, food specially designed for patients with cardiovascular diseases. So classification helps each category of people to choose herbs wisely. So when you want to prepare a food, you can choose herbs or you can choose food which also has a therapeutic use. Scientific study, classification based on active constituent, very important when we are looking at the technical part or scientific part of herbs. Each herb is made of diverse group of constituents. So when we are aiming a particular therapeutic medicine for a disease, and when we know that it is linked to a particular secondary metabolite present in the plant, we can look at classification of herbs having these secondary metabolites. So classification is based on the active constituents. For example, plants having alkaloids, plants having essential oil, or simply some trace elements or minerals, salts, which helps the pharmacologist and herbalists to understand how or where or which receptor the herb is going to act in the body. So for example, bitters, if chosen, help in detoxification, while as all the aromatic plants, they stimulate digestion. Further classification like the life cycle management, where herbs are classified as annual, biannual, perennial. When we know the life cycle of a plant, then cultivation becomes easy. Cultivation is a part of herbal drug industry where there is a continuous supply of these herbs to the industry. So when this cultivation is planned, it is best to know the life cycle of a plant. Hence, a cultivator can economically and efficiently plan the cultivation of medicinal plants. Earlier, a lot of them were collected from wild sources, but these days medicinal plants are cultivated for a purpose where quality can be built in during cultivation. Another advantage of classification is there is preservation of knowledge. When we organize herbs into categories, this ensures that the traditional and modern knowledge is preserved and is passed on to generation to avoid confusion between similar looking plants. Many times too many plants are given the same word. For example, when we say rasna, there are different types of rasna. So when we classify them botanically and therapeutically, it will be easier for us to choose a particular herb. Most important, classification is necessary for safety and accuracy. When we classify and when we say that these group of herbs are classified and are known to be toxic, one can use them very carefully when incorporated into a formulation. This is the first example where herbs are classified for therapeutic action. For example, all the carminatives, if you look at this, for example, fennel, peppermint, coriander, caraway, ajwain, clove, all these which have smell and essence or a typical flavor, these are used as carminatives. They help in relieving gas from the digestive system. Drugs like senna and aloe, which are known to contain anthraquinone glycosides, are used as laxatives and purgatives, and they help in relieving constipation. So when we are targeting a particular formulation for these uses, you can look at these class of drugs. Expectorants, drugs like licorice and vasaka. Vasaka, we all have heard of adulsa. Adulsa is a syrup which contains vasaka leaf extract. So these are classified as expectorants. There are some antiseptics. Age old turmeric, which has stood the test of time, is a classic antiseptic and helps in prevention of infection topically and internally. There are very good adaptogens. So these days when we are looking at overcoming stress and we want to choose a herb, drugs or herbs like ashwagandha and ginseng are excellent adaptogen and they help in regeneration or overcoming stress. Another classification which is based on the botanical or morphological features is dicots, for example, neem and digitalis, monocots, for example, aloe and garlic. So, this class of herbs is important when we are authenticating and establishing an identity for a herb. So, the way we recognize human beings by their features, here we can recognize the plants by looking at their external morphological features. Gymnosperms are flowering plants, so example is ephedra. Pteridophytes, for example, Lycopodium spores. Fungi and lichens, example, yeast and Eusenia. Based on the habitat, very important, when we are trying to collect a herb, we should know the exact habitat. Now this habitat helps us not only to search the drug, but these days when you are collecting the herb, you need permissions. Suppose the herb is collected from a wild source, wild herbs, and you have to enter a forest, a permission from the forest department is very essential, and there is a mandatory requirement of having certain group members when you go in the forest and collect these herbs. So, without the permission of the forest department, you cannot enter the forest and start collecting herbs. So, knowing the habitat is very important. Wild herbs, cultivated herbs, aquatic herb. So, when you want to collect drugs like hydrocotyle, agar, lotus, we must know or we must understand that this is from marine source and necessary arrangements like the boat, fisherman, net needs to be done before we collect herbs. Desert herbs, aloe, commiphora, these are desert plants or desert herbs. Mountain herbs, rhodiola or cordyceps. So, when we know these habitats, it is easier to prepare for the herb collection. Two major sources of herb is, one is the wild source and the other is the cultivated source. Wild source is naturally occurring habitat of the drugs, and major of them are the forest, mountains, valleys or aquatic sources. Cultivated is when an industry wants a continuous supply of herbs, it is better to cultivate the herbs. Let us first look at the wild sources. Herbs are collected directly from their natural habitats such as forests, grasslands, mountains and plains. Example, Digitalis purpurea, Rolfia serpentina, Nux vomica, Terminalia chebula and Saraka indica. Of course, because of the demand of these herbs, some of these are now being cultivated. For example, Digitalis which contains cardiac glycoside, and there is enough scientific proof being generated for digitalis to be very effective on a failing heart. Hence, the demand for digitalis has gone up and this is now being cultivated. Similarly, for Rolfia, many parts of India now cultivates Rolfia. Initially, it was obtained from wild sources. Saraka indica is the Ashoka plant where Sita was known to kept captive. So, this is the Ashoka Vatika where these plants are found in abundance. This is a huge tree and still collected from wild sources. There are few very good advantages when herbs are collected from wild sources. Plants are collected from their natural habitat and a natural environment. So, hence they possess high potency and authentic phytochemical profiles. The original active constituents are known to be present in these plants. There is no need to spend or take extra efforts to add fertilizers and pesticides when herbs are collected from wild sources. Hence, as compared to cultivated, there is a low cost of raw material. there is greater genetic diversity which helps in discovering new bioactive compounds, and collection is ideal for rare and endangered herbs which cannot be successfully cultivated because they grow at a particular latitude or longitude. Especially if we look at many herbs which grow in Himalayas, these conditions cannot be regenerated when we want to cultivate them. However, there are few limitations: variation in quality due to uncontrolled environmental condition. Nature is not in our hands, so if there are sudden climatic changes which can cause a change in the active constituent, that becomes a limitation, and hence batch to batch variation can happen when we collect drugs from wild sources. There is a risk of over exploitation and extinction which we have faced. Sandalwood tree is a classic example where it was over exploited, and hence now there are government rules on collection of sandalwood. Lack of traceability and sustainability issues. Every time you do not know which part of the jungle this herb is growing. Today if you have collected from a particular region and if you have exhausted that herb, next time you have to go searching where this plant grows. Of course, the tribal people are aware of these sources, but you cannot totally rely on this. As compared to wild source, a cultivated source is much more reliable. You know where the herb is growing, you know at what time the harvest is ready, and a few examples are aloe vera, which is being cultivated in large amounts because there is a big demand as a therapeutic agent, as a cosmetic, as a nutraceutical. Hence, aloe these days is being cultivated. Vidhania somnifera, which is ashwagandha, especially after the pandemic where there is an awareness of wellness, there is a big demand for ashwagandha, and ashwagandha is also now being collected in fields. Mentha arvensis, which is greatly demanded by the food industry as a flavor and also for therapeutic uses. Curcuma longa, this is an age old Indian traditional medicine which has a large use as wellness, nutraceutical, therapeutic herb used topically and internally. Cassia angustifolia, which is senna, which is used as a natural laxative. So, in these days where there is a change in lifestyle and when enough fibers are not being consumed, drugs like isabgol, senna are in demand as natural laxatives. So, these are being cultivated. Looking at the advantages of cultivated courses, these are more standardized. We can rely on them for a uniform quality. There is a control over soil, water, fertilizers and harvesting. There is sustainability and traceable supply. You know from where to collect these herbs, and there is a regular supply to the industry, which means that there is batch to batch control and less of batch to batch variation. It is easier to implement good agricultural and collection practices when drugs are being cultivated. Enough of pharmacovigilance can happen when drugs are cultivated. Possible to select and propagate high yielding varieties. So, when you select seeds of good quality, good genetic makeup, you can expect a good yield. Helps in conservation of wild species by reducing the wild harvesting pressure. However, there are a few limitations. The high cost as compared to wild source, because you are investing in land, you are investing in fertilizer, you are investing in physical labor, or you are investing sometimes in some special equipments which are used for harvesting and drying. Reduced genetic diversity as compared to wild plants, this happens because you are selecting a particular kind of seed. So, you expect only those active constituents to be present depending on the genetic makeup of the seed. And If the seeds are mutated or if the DNA genetic changes have occurred, then this is expected in the final crop. Some species lose potency when grown outside their natural habitat. Very, very important, when a plant is collected from its natural habitat, it thrives in its natural habitat. So, when you try to plant it somewhere, when the latitude, longitude changes, maybe there are changes in the active constituent. And this is seen in literature. When you collect turva from Maharashtra and Karnataka, the constituents vary. That is what happens when we change the geographical source of a drug. All the umbelliferous fruits containing volatile oil best grow in the state of Gujarat. So, this is what that they may lose little potency if the habitat has changed. Other than the wild and cultivated sources, there are some other sources from where herbs or their constituents are obtained these days. Tissue culture, this is a test tube garden where herbs are produced by micro propagation, and we can expect disease free plants. A biggest advantage here is there is no need to invest in land, and we need not wait till the plant grows fully. Tissue culture is a separate technique, and here we can harvest the cells for their active constituent. For example, stevia, which is a natural sweetener and for which there is a big demand, has been propagated through tissue culture. Marine sources, as I mentioned earlier, algae, seaweeds and other marine herbs which have known to exhibit anti-cancer, anti-hypertensive and useful for different cardiovascular diseases or useful as antibiotics, are now being cultivated or obtained from marine sources. The only disadvantage is we have to dive deep sometimes, or special people are needed for collection from marine sources. Here also permissions are very, very important when one has to enter the sea or ocean for collection of herbs. Greenhouse or hydroponic cultivation, where herbs are cultivated in controlled environment, and these are for sensitive or high value herbs. For example, basil, thyme, ginseng, for which there is a big demand today. So, summarizing, from wild sources, digitalis was known to be collected from wild sources. Digitalis and Rauwolfia were known to be found out from wild sources. Cultivated sources, we find a lot of aloe vera, we find a lot of papaya being cultivated these days for medicinal properties or for the enzyme papain, Vidania somnifera. These are just a few examples, but there are large number of examples where drugs are being cultivated today. Amla is being cultivated, Safed Musli is being cultivated, Triphala, all the three ingredients because of the demand is being cultivated. Tissue culture, the best example is stevia. Marine sources, spirulina, agar remain to be collected from marine sources, and the greenhouse source, basil, is known to be commercially collected from greenhouse cultivation. Why is it important to know the source? It is important to know the source so that we can ensure quality, safety, efficacy and sustainability, and to a certain extent we can ensure that no adulteration or no substitution drugs are being collected. So, to ensure the correct identification and authenticity, it is very important that we look at the morphology when we are collecting the herb. Collecting from wrong region or habitat can lead to misidentification or adulteration. For example, Digitalis lanata and Digitalis purpurea, though both species of Digitalis are known to be potent as cardiotonics, collecting them from different sources can lead to different identification of species. to maintain consistent chemical composition. So, if you collect from different regions, maybe there is a difference in the active constituent. So, collecting herbs for their optimum active constituents, it is better to restrict to a particular geographical habitat or origin. For example, Rolfia serpentina when collected from different states showed difference in the rhizopene content. To determine the right time and part of collection, it is important to know the source, which part of the plant is collected, whether it is the root, the leaf, the bark. So, when we are looking at Vasaka, Vinca, it is the leaf. When we are looking at cinnamon, cinchona, wild cherry, Arjuna, Ashoka, the bark is collected. When we are looking at flowers, clove has to be collected as a flower bud and not allowed to bloom. Different types of seeds like sesame, mustard, almond, the seeds are collected, and the season of maturity. Barks are collected after the rains in autumn when the sap is rising. Leaves are collected just before the flower forms a fruit. If leaves are collected when fruits are formed, then they are little matured leaves. So, this ensures a maximum concentration of active constituent. For example, peppermint leaves are collected just before flowering. This ensures the maximum oil content. To avoid contamination and adulteration, plants should be avoided to be collected from roadsides because there are chances of heavy metals or pesticides or toxins. So, the FDA has come down very strictly on identification for any wellness, nutraceutical or food product which is put in the market containing a herb, that they should be free from heavy metal, pesticide or toxins. So, when plants are collected, for example, if they are grown in an habitat where dirty water is there, they absorb these heavy metals and toxins. So, to ensure safety and purity, it is best to collect from a good cultivated or a wild source. For standardization and quality control also it is important because there are reproducible results and good agricultural and collection practices can be applied. It is also important for sustainable and ethical sourcing. Over harvesting from wild sources may cause ecological imbalance and endangerment of species. Most important, to meet the regulatory and pharmacopoeial standards which are mentioned in different pharmacopoeias for different kinds of herbs today. So, to summarize, authenticity is very important to ensure the correct plant species, to maintain the potency, safety for standardization, sustainability and regulatory compliance. What are the different kind of herbal drug preparations, or we have just seen different sources of herbs, what is a herb. Now, where this herb enters in commercial market, how is this herb present? For example, herbal drugs can be prepared or they are present in various forms. Let us have a quick look at this. We are going to discuss in the next unit what are the traditional herbal preparations. Here I am just going to introduce to you what are these preparations. So, herbs are a part of traditional Ayurvedic preparations. For example, asavas, arishtas, gutikas, vatikas, gritas, where they contain herbal extracts or the herb powder. The basic form in which a herb can be presented or prepared for the market is the raw form. This is the unprocessed form where a herb is collected, dried and powdered. For example, dry ginger, dry senna leaves, dry licorice root powder. So, this is the basic raw form, and from here a formulator decides as to how to further process it. This is the most simplest preparation of a herb with the lowest cost and minimal processing, where it retains all the constituents. So, one can decide if you have to separate a particular constituent from this or use the entire herb powder when you want synergetic action of all the active constituents present in it. Powders and churnas. Herbs are powdered and a mixture of these powders is called churnas. So, this can be used as such. Infusions, where herbs are added to warm water and they are present as infusion. For example, there is peppermint infusion or chamomile infusion. So, sometimes these dry herb powder are supplied and you can make an infusion by just immersing it in hot water. Decoction are concentrated liquids where a herb is boiled in the solvent. Dashamula Kwatha or ginger or tulsi decoctions for cold. We are going to discuss this later unit. Tinctures, where herbs are macerated in hydroalcoholic solutions. These are extracts. This is another form of herb most commonly occurring in the market and too much in demand. So, these extracts can directly form part of any formulation that a formulator wants to do. Oils and resins are extracted from a herb from the herb matrix, and syrups and elixirs. These herb powder or the extract then goes on to form a pill, tablet or a capsule. If it is a topical formulation, these herb extracts are formulated as ointment, cream or a gel. So, these are different types of herb preparations that are present commercially in the market when we want to include a herb. So, an herb can form the main therapeutic ingredient, or most of the times it can also act as an excipient. So, in syrups, in creams or in ointments, when we use acacia and tragacanth, they can act as a excipient also. These are asavas and aristas. These are naturally generated alcoholic preparations. Other modern forms: lozenges, herbal teas, essential oils and herbal cosmetics. So, these are some of the herbs and their forms which occur. For example, crude drug, the ginger powder. There are many examples which we cannot discuss on one slide, but these are just few examples where herbs and their types are given here with suitable uses. Thank you.